So today I have the honor to introduce you to the phenomenon of split open reading frames. And now I guess that you're all wondering what a split open reading frame is. So let's start by explaining that. In the upper part of this figure, you see a very simplified schematic of a protein coding transcript. We have the 5' untranslated region, an open reading frame that encodes the protein product, as well as the 3' untranslated region. And now, for example, splicing alterations can introduce an extra exon into the transcript. And if this extra exon introduces a premature termination codon into the open reading frame, we have a truncated open reading frame. Normally, transcripts with a truncated open reading frame will get degraded by nonsense-mediated decay. But this is not always the case. And if this transcript persists, then this truncated open reading frame can get translated into a partial protein product. And if further downstream there is another in-frame start codon, there is a second truncated open reading frame that can also be translated into a partial protein product. Okay.
I think it's out of my control, but sorry for the inconvenience. I cannot do anything, so we can continue and maybe help will come to assist on that. Sorry.
Okay, so we were at this point where this transcript with the premature termination codon gets translated, and as we have the two truncated open reading frames, we get two partial proteins. And these split the original full-length protein into pieces. And this phenomenon is termed a split open reading frame. And split open reading frames were first observed for a splicing factor called SRSF7. And SRSF7 is involved in the splicing of many essential genes, such as clock genes. And it was seen that when SRSF7 is overexpressed, it binds to its own poison cassette-containing transcript and stabilizes it. And upon the stabilization, the two truncated open reading frames can be translated into two proteins, which both serve different roles. So the first protein is in direct competition with the full-length SRSF7 protein because it has the same binding domain. However, it lacks the functional domain for spliceosomal assembly. And the second protein renders the transcript NMD resistant. And from this finding, we went on to ask ourselves whether split ORFs are an exclusive phenomenon to SRSF7 or whether they are observed more widespread. And in order to answer this question, we developed the split ORFs pipeline. And the idea of the split ORFs pipeline is that it predicts whether a transcript could potentially form a split open reading frame based on the transcript sequence. And this is due to two criteria. First of all, the transcript needs to have at least two open reading frames. And second of all, both of these open reading frames need to align to the same target sequence. And with target sequence, I mean the full-length open reading frame that is split into pieces. And now the split ORFs pipeline works in 3 steps. First of all, we find all possible open reading frames. Then we identify unique regions of the split open reading frames, which we can later use for validation. And third of all, we find the target transcript. So let's have a closer look at how this works. In the first step, we have the transcript sequence, and we simply note down all possible open reading frames by going through all of the 3 reading frames of the transcript. And as an open reading frame, we only consider canonical AUGs, and then we stop at the first in-frame stop codon. And all of the start codons that are in frame and within the open reading frame are disregarded. So we only consider the longest open reading frame. And in this example here, we found 2 open reading frames. In the first, there is none in reading frame 2, and there's one in reading frame 3. And in the next step, we want to identify unique regions of these possible open reading frames. And for this, we perform an exact matching to a protein coding reference. And the protein coding reference are all transcripts as annotated in Ensemble to be protein coding and quality filtered for transcript support level 1 and 2. And we use a tool called MAMR3 to perform exact matching. And here in our example, you can see that the open reading frames in reading frame 1 match exactly, so here in light green, to the protein coding reference. And in reading frame 3, there is no exact match found. And this information we can use to determine the unique regions, because these yellow regions here that did not match exactly to any of the protein coding transcripts, these are unique to the split open reading frames. And we do this for both DNA sequences and protein sequences, because we can use the unique DNA regions for validation with ribosec data and the unique protein regions for validation with mass spec data. And the last step of the split-offs pipeline is to find the target transcript. So we now take the regions that did match exactly, and we blast them against the protein coding reference. And this we can use to identify the potential split ORFs, because a split ORF needs to have at least 2 open reading frames, and both of these open reading frames need to align to the same target sequence. Okay, so this is how the split-offs pipeline works, and we applied it to 2 sets of transcripts that are annotated in Ensemble. The retained intron transcripts, because there's already some splicing variation going on, as well as the nonsense-mediated decay transcripts, which are especially interesting because they already contain a premature termination codon and hence are very good candidates to form split open reading frames. And as you can see, for both sets, we predicted quite some thousands of transcripts together with their unique DNA and protein regions. And in the following, I will focus on the results for the nonsense-mediated decay transcripts. So first of all, we performed a geoterms analysis for the genes associated to the transcripts that are NMD and predicted to form split open reading frames. And in general, there were a lot of RNA, DNA, and protein interaction or binding terms. And there were also some terms involved in mRNA processing, mRNA stability, and also the spliceosomal complex. And the next thing is that we want to validate the predictions that we made with the split-offs pipeline. And for this, our first idea was to use a model that is much more complex than our rule-based, rather simple approach. So we took Translation AI, which was published this year, and it predicts the probability of translation initiation sites given the transcript sequence. And so our idea was to give Translation AI our split-ORFs transcripts, which have at least 2 annotated start sites, because we always have at least 2 open reading frames, and see how high of a probability for translation initiation sites these get. And we decided to make 2 sets out of them. So we have the best split-ORF start, which is the start site in the split-ORF transcript with the highest probability. In this example, this would be start 1 with a probability of 90%, and the second best split-ORF start, which is the start site with the second highest probability. So in this case, start 2 with a probability of 20%. And of course, we need to compare this to a background. And as a background, we decided to take the NMD transcripts that are not predicted to make split open reading frames. And for these transcripts, we also wanted to have the best and the second best start. But for these, we simply took all of the start sites within the transcript and then took the one with the highest probability as the best non-split-off start, which is here start 1. And the second best non-split-off start is then the start with the second highest probability. So here, start 3. Okay, so now we can compare these sets of start sites. And here on the left, you see a density plot of the probability of the best start sites, in blue for the split-off transcripts, and in red for the non-split-off transcripts. And we can clearly see that the split-off transcripts tend to have higher probabilities for the best start sites. This is also significant. But what is even much more interesting are the second best start sites. Because if you think about NMD, NMD is a translation-dependent mechanism, so one start site needs to be used anyway. However, thinking about split-orf, there are two start sites that need to be used. So looking at the probability of the second best start site is much more characteristic for a split open reading frame. And here on the right, we have the density plot of the second best start site probabilities, again in blue for the split-orfs and in red for the non-split-orfs. And again, you can see that the split-orfs tend to have much higher probabilities for the second best start sites, and this is also significant. However, in general, the probabilities are much lower, and also for the split-orfs, we see quite some second best start probabilities close to zero. Now, this doesn't worry me too much, because out of these thousands of transcripts that we predicted to form split open reading frames, well, there can well be quite some false positives. Another reason for this might be, if you think back about this example of SRSF7, the second split-orf was translated less frequently. So it can also be that the second best start sites are not used as frequently. Hence, they have a less strong context and also get a lower probability. Okay, so this was already quite a promising result for our predictions, but of course, we next need to look into data and see whether we can validate our split open reading frames. And for this, we took ribosec data of different cell types. So we have cardiomyocytes derived from induced pluripotent stem cells, as well as human umbilical vein endothelial cells, or HUVECs. Both cell types are involved in the cardiovascular system and also very relevant to cardiovascular disease. And we also have a cancer ribosec data set of leukemic white blood cells. And our idea for the ribosec data was to use these unique regions as predicted with our split-orfs pipeline, and to see whether we can find ribosec coverage in these unique regions. So what we did was we took the ribosec reads and aligned them to the genome, and then we intersected with the unique regions as predicted by the split-orfs pipeline. And if you remember, these unique regions were sequences that are not present in any other protein coding transcript. So these are characteristic for the split-orfs. And then these intersections we normalized by length. And now the question is, of course, how much coverage in a unique region do I need to say that the split-orf is validated? And for this, we calculated a background. And we took regions that we believe are not translated. So we took 3' untranslated regions of protein coding transcripts as annotated in Ensemble. And from these regions, we sampled regions of the same length as the unique regions in order to get the same length distribution. And then we performed the same steps. So we intersected with the ribosec reads, and we normalized this by length. And from this, we got a background distribution, and we took the 95th quantile as a threshold and said, if we observe more coverage in a unique region than this 95th quantile of the background, then our unique region is validated. And now I, of course, want to show you the results of this pipeline for our data sets that I introduced. So you can see that for the HUVECs, more than 500 unique regions were validated with this approach, a little bit less than 200 for the cardiomyocytes, and 144 for the leukemia cells. And some of the unique regions are only validated in one cell type, while others are validated across cell types. And just to give you a feeling of how much we validated here, taking all of the unique regions that the pipeline predicted, this is a validation rate of 11.72%. And this is, of course, already a nice hint that split-orfs might indeed not be exclusive to SRSF7, but are more widespread. And in order to pursue this further, I will come to my outlook, because we have experiments planned where we will get ribosec and mass spec data of NMD-inhibited UVEX and cardiomyocytes. And this will be really interesting, because if we inhibit NMD, the transcripts with the premature termination codon will be stabilized. And thinking back about this original publication about SR7, the transcript with the premature termination codon also first needed to be stabilized to be then translated. And for this, we will also have long reads, of which I already performed the assembly, so I can give you a little teaser here. And the idea is, of course, to detect novel and cell type specific isoforms, hopefully also NMD and split-off isoforms. And for the cardiomyocyte assembly, we have more than 6,000 novel NMD transcripts, and more than 4,000 are predicted to make split open reading frames. And for the UVEX, there are more than 3,000 novel NMD transcripts, and more than 2,000 are predicted to make split open reading frames. And with that, I would like to thank a couple of people, especially Marcel Schulz, who's been a great guidance in this project, as well as our collaboration partners and the funding. And I thank you very much for your attention, and I'm open to take any questions.
So questions. So how do these split-offs actually work functionally? Is it that the translation machinery sort of translates the first ORF, then folds off and sort of rebinds maybe even another translation machinery molecule, meaning you would have a second Kozak sequence? Or does it sort of stop translation but then keep scanning? Do we know that, or?
The answer is we do not know that, but we are, of course, hardly thinking about this as well. This is one of the big questions. Yeah, I mean, the mechanism that you just outlined are pretty much the possibilities that we have in mind as well. And yeah, I think this is something we will also investigate in the future, but for now I have to tell you, we also don't know.
So maybe one way of looking at this is to understand what translation AI actually takes into account, you know, to predict the translation. Because if it's some sequence things, and if it looks at a Kozak sequence, right, then that would be probably what is there. Otherwise, it wouldn't have predicted its translation. Or if it is purely based on mass spec data, then maybe it doesn't. I don't know.
Yeah, so translation AI is a sequence-based model, so the Kozak sequence definitely should play a role, but I think also other sequence features, but that's a very good point. Thank you very much. We will look into this.
Great talk. I wanted to ask you, we're expecting to find your split ORFs in all the cell types that you've looked at, but don't you think that it's something that only happens in a certain stage, let's say, of the cell, or in some tissues and not others? Definitely if you're looking at intron retention, right? So only when there's intron retention will you get the split ORFs. And then you shouldn't expect to find it in all the data sets that you're looking at, right?
Yeah, I would definitely agree. So what I didn't explain is that in these data sets that I looked at, for example, for the UVEX, we also had different conditions. So we had normoxia and hypoxia. So I would definitely expect that depending on the condition of the cell and also of the cell type, that we will find different split ORFs, or maybe in some more and in some less. And this is also the motivation of these new data sets where we performed a long read assembly to really see which isoforms are cell type specific and which of them form split open reading frames. Yes.
Hello, yeah, thank you for presenting this really novel research. My question is probably more technical. If you go back to the slide where you try to find the unique regions, it looks like the unique regions are the regions missing in this split ORFs. How finding them would prove splitting ORFs actually exist? I kind of didn't quite catch the logic there.
Yeah, so these regions are not missing in the split open reading frames. They are part of the split open reading frames. They just do not have sequence identity with any of the protein coding transcripts that are annotated. And our reasoning behind this is, if you want to validate this whole split ORF, and you look into ribosec data and you see a lot of coverage, then you're happy, right? You validated the split off, but maybe this is just because it shares a part of the sequence with the protein coding transcript. So maybe what we see then is coverage in the protein coding transcript, which shares a sequence with the split offs. So this was the motivation behind it, that we find the regions that are not shared with any of the protein coding transcripts, so that if we do see coverage, we can be sure that this is due to the split offs and, well, yeah, not from a other protein coding transcript.
Okay. Okay. So thank you so much, Christina. Let's thank her again.